and cheek cells. The cell is the basic structural and functional unit of all known living organisms. Some organisms, such as bacteria, are unicellular, consisting of a single cell. Other organisms, such as humans, are multicellular, having many cells. A cell mainly consists of a fluid-filled space called cytoplasm and is surrounded by a cell membrane. The cytoplasm contains a nucleus, mitochondria, Golgi bodies, lysosomes, endoplasmic reticulum, ribosomes, and vacuoles. Plants have an additional boundary wall called the cell wall, which is absent in animal cells. Plant cells have large liquid-filled vacuoles, which occupy most of the volume in the mature plant cell. Plant cells also contain chloroplasts, which is absent in animals. To prepare a stained temporary mount of an onion peel. To do the experiment, we require cover slip, glycerine, glass slides, onion, watch glasses, distilled water, saffron in solution, forceps, brush, needle, compound microscope, blotting paper, and a dropper. Procedure. Pour some distilled water from a bottle into a watch glass. Take half a piece of onion and remove leaves from the onion piece. Remove a piece of transparent onion peel with the forceps. Put the epidermis into a watch glass containing distilled water. Take some saffron in solution using a dropper. Put a few drops of saffron in solution into another watch glass. Using a brush, transfer the peel into the watch glass containing saffron in solution. Keep the epidermis for 30 seconds in saffron in solution so the peel can become stained. Take the peel from the saffron in solution using the brush and put it in the watch glass containing distilled water. Using a dropper, take some glycerine and put 2 to 3 drops of glycerine in the middle of a dry glass slide. Transfer the peel to the slide containing glycerine using the brush. Take a cover slip and place it gently on the peel with the help of a needle. Remove the extra glycerine using a blotting paper. Take the glass slide and place it under the compound microscope. View the slide under the compound microscope. Observation. A large number of regularly shaped cells are lying side by side. Each cell has a distinct cell wall. A distinct nucleus is present on the periphery of each cell. Lightly stained cytoplasm is observed in each cell. A large vacuole is present in the center of each cell, surrounded by the cytoplasm. Conclusion. As cell walls and large vacuoles are clearly observed in all cells, the cells in the observations are plant cells. Precaution. Use a brush to transfer peel from one apparatus to another. Staining of the peel should neither be too dark nor too light. Extra glycerine stain should be removed by using blotting paper. To prepare a stained temporary mount of human cheek cells. To do the experiment, we require toothpick, cover slip, methylene blue solution, glycerine, glass slide, distilled water, brush, needle, compound microscope, filter paper, and a dropper. Procedure. Pour some distilled water from a bottle in the middle of a dry glass slide. Take a clean toothpick and gently scrape the inner side of the cheek. Rub the cheek tissue on a glass slide that contains water. Mix the mixture using a needle and spread it. Take methylene blue solution using a dropper and add a few drops of it to the mixture on the slide. After 2 to 3 minutes, remove excess water and stain using a blotting paper. Take glycerine using a dropper and add a few drops of glycerine to the test mixture. Take a clean cover slip and lower it carefully on the mixture using a needle. Using a brush and needle, press the cover slip gently to spread the epithelial cells. Remove extra liquid around the cover slip using a blotting paper. Take the glass slide and place it under a compound microscope. View the slide under the compound microscope. Observation. A large number of flat and irregular shaped cells are observed. The cells do not have a cell wall. However, each cell has a thin cell membrane. A deeply stained nucleus is observed in the center of each cell. No prominent vacuole is observed in the cells. Conclusion. If the cells observed do not have cell walls or prominent vacuoles, the cells of the specimen on the slide are animal cells. Precaution. Ensure toothpick used to scrape the cheek is clean so as to not cause infection to the cheek. Extra stains should be removed by using blotting paper.
So here now we are, after observing the onion cell and human cheek cell, let us compare a typical plant and animal cell. As you can see from the figure itself, animal cell is more irregular, whereas a plant cell is more geometrical in shape, and there is the thick outermost covering, जिसको हम कहते हैं cell wall, जो rigid है, non-living है, whereas in case of animal cell there is no cell wall, there is a thin outer covering which is called cell membrane or plasma membrane. और ये cell membrane या plasma membrane plant cell में भी present है, but it is inner to the cell wall के अंदर है, ठीक है? So they are getting a double protection, more protection, plant cell. Then you can see a nucleus, a prominent nucleus here right in the center of the animal cell, whereas this nucleus is pushed to a side or periphery in case of a plant cell. Why is it pushed to the side? Is because it is having a vacuole, a central large vacuole, which is pushing it to the side. This vacuole is actually occupying nearly all the space in the plant cell, which is large, and that is why it is pushing the nucleus to the side. But in case of an animal cell, vacuoles are generally absent, but if they are present also, they're very small in size. In case of plant cell, there may be 1 or 2 vacuoles, which are very big in size. Then you will see these colored bodies, which are called as chloroplast in case of a plant cell, but it is absent in case of an animal cell. Chloroplast ka role jo hai, it is a colored plastid, a green colored plastid, which contains chlorophyll in it. And this chlorophyll is an essential component which absorbs the sunlight and is responsible for bringing out photosynthesis process in case of plant cell. Since it is absent in animal cells, so animal cells are not able to prepare their own food, and hence it is seen only in plant cell and not in animal cell. Then we have mitochondria. Mitochondria are powerhouse of the cell, which are present in both. All right. So there are Golgi apparatus, there are endoplasmic reticulum, there are a whole lot of cell organelles which are present both in plant and animal cell. तो आपने ये basic, इसमें there are large number of cell organelles which are shown, but at your level you need to do only this much of comparison, which is the diagram which is given even in your book on page number 96. A typical animal cell will have a nucleus which is present essentially in the center, whereas in plant cell it is present on the periphery. And then you have a cytoplasm. Cytoplasm is common to both plant and animal cell. It is the jelly-like substance which is present between the cell membrane and the nucleus. ध्यान से देखिए, this blue part which is present between the cell membrane and the nucleus के बीच में जो space है वो occupy कर रहा है cytoplasm. Then there is a thin cell membrane which is protective, which is present, which is the outermost covering in case of an animal cell, but it is the second most outermost covering in case of a plant cell, because plant cell has cell wall as another protective covering which is present outer to the cell membrane. So we have mitochondria which are common to both, which are called as the powerhouse of the cell. And then there are chloroplast, which are, these are green color plastid, which are present exclusively in plant cell and absent in animal cell. And then we have a large vacuole, which is present only in case of plant cell. Animal cells may generally absent hote hain, but if they are present, they are small and large in number. In case of plant cell, they are large, but they are small in number, maybe just one or two. So coming to the comparison, a plant cell is usually large and an animal cell is comparatively small in size. हम सारे cells of animals के सभी cell को generalize कर रहे हैं, plant cells को generalize कर रहे हैं. हमने variety of cells के structure देखे हैं और आगे भी हम लोग higher classes में भी पढ़ेंगे, लेकिन हम एक generalize कर रहे हैं कि एक typical plant cell generally large होता है size में when compared to an animal cell. Then cell wall is present in case of an animal's plant cell, but it is absent in an animal cell. Chloroplast, जो green color plastids होते हैं, they are present in plant cell as you have seen in the diagram, but they are absent in animal cells. Nucleus जो होता है, it is present in the periphery in case of plant cell, but it is present almost in the center in case of a animal cell. Centrioles are another bodies, microtubules होते हैं, which are present in case of animals alone. ये animal cells में obtain देखे गए हैं, but they are absent in case of a plant cell. We'll be studying about centrioles and many more cell organelles like mitochondria, chloroplast, Golgi apparatus, lysosomes, all these in higher classes.